The overall goal of this procedure is to acquire knowledge of the technique of electrophoresis to compare the migration of DNA with different concentrations of agarose and to determine the quality of various samples of DNA.
The electrophoresis is a method of separation of biomolecules fundamented in the electric charges that they possess. Some examples of its applications are checking the quality of DNA, verifying the presence of an insert in a plasmid, confirming the identity of a suspected being, figuring out the homozygosity or heterozygosity of some genes that have different cleavage sites in each case. The reactives and running conditions may vary with the biomolecule used. For RNA DNA, we would have a matrix composition usually of agarose, even though it depends on the sample size and the objective. For proteins, usually polyacrylamide yields are used. Talking about the running buffer, for DNA we have the TBE, or we also have TAE. For RNA we have the MOPS buffer. For proteins we have the trisglycine SDS. The most used loading buffer for DNA is the one that contains 0.25% of blue of bromophenol, 0.25 of chelancianol, and 30% of glycerol. The one for RNA contains the same reactives, but also contains EDTA and formaldehyde, in order to avoid secondary structures. For proteins, we have special buffers to unfold and to give them a negative charge, such as the 4X protein loading diet. Examples of ladders, in the case of DNA, would be the 1 kilobase DNA ladder from in vitrogen. For RNA, we have the 0.1 up to 2 kilobase RNA ladder ambient. For proteins, we have the supercyclic molecular weight protein ladder from Thermoscientific.
Each team was engaged of preparing different types of agarose concentrations as well as the chosen running buffer. Prepare agarose plus the buffer solution. Dissolve the solution by adding heat with the microwave until we get a homogeneous mixture. Cool the solution to 65 Celsius degrees approximately and add intercalating agent to visualize DNA and RNA bands. Pour the solution into the gel carrier plate. Wells are made with a comb that was previously placed in the gel carrier plate. Wait until it reaches a solid composition. Put the gel in the electrophoresis chamber and add the appropriate running buffer. Add the DNA samples with the loading buffer into the wells with a micropipette. Apply voltage with a power supply. Wait until colorants arrive to the known positions to stop. Visualize the DNA, RNA, protein fragments in a transilluminator.
DNA electrophoresis with different agarose concentrations in a TBE buffer and a TAE buffer. Regarding nucleic acid samples visualization in a 0.7% agarose gel, there's a protein contamination in the fourth well as well as plasmidic DNA degradation. In wells 2 and 3, there is an excess in the amplified fragments and genomic DNA degradations respectively. In the 1.5% agarose gel, there are an excess in the amplified fragment, a genomic DNA degradation, protein contamination, and an excess in loading buffer. In the 2.5% agarose gel, it can be seen two protein contamination, that one in the DNA layers well and the plasmidic DNA well. There are two DNA degradations, genomic DNA and plasmidic DNA. Finally, in the 1.5% agarose gel, there are two DNA degradations as well as a low level of unspecific DNA amplification.
When quality is not visualized, the authoradiograph will show the following results. DNA RNAs with salt excess. That happens because there's an error in the lysis method altogether with the separation of genetic material from other cellular components. Some detergents are composed of salts and those are not well separated, causing impurity in the next steps. And the purification of nucleic acids when using salts and ethanol to precipitate DNA RNA. DNA RNA degradation. This is due to an incorrect lysis methodology. Besides that, can be degraded when not stored properly. Excess amounts of intercalating agents, contamination with protein, and excess amounts of loading buffers.
HEL-RED is an intercalating agent in order to visualize nucleic acids through the transilluminator. Its usage is preferable rather than other intercalating agents such as ethidium bromide because it is less dangerous, less mutagenic, and has a high degree of sensitivity.
Choosing the agarose polyacrylamide percentage depends on the length of the genetic material. In this case, the 4 teams of the genetics engineering laboratory made 3 types of agarose gel compositions: 0.7, 1.5, 2.5% in order to separate, identify, and purify samples. All the sample bands analyzed in a 0.7% agarose gel were clearly seen due to the migration freedom. It's recommendable to analyze fragments whose length takes values between 800 base pairs to 12,000 base pairs approximately, whereas Two samples bands analyzing a 2.5 agarose gel couldn't be seen as independently due to the lack of migration freedom. The buffer selection will depend on four basic conditions: salt concentration, regulatory capacity, the running time, and voltage. In this case, 3 of the 4 teams use TBE running buffer. The TBE buffer is often used in gel electrophoresis when there is the need to apply high voltages due to its high concentration of ions. Thus, its regulatory capacity is better than TIE buffer. Besides that, TBE buffer can be applied for long-running processes rather than TIE buffer. Loading buffers are widely used for the following reasons: increasing nucleic acids density, visualizing nucleic acids loading, and a source of running processes. When carrying out electrophoresis, it is important to consider the electrode's distance given by the size of the electrophoresis chamber. In this case, the distance was 20 centimeters. Then, according to the DNA size of the samples, it was determined that a voltage of 1 to 5 volts per centimeter will be applied, giving an approximately final voltage between 100 to 130 volts. Now, it's important to remember when using high voltages, DNA samples will migrate much faster than low voltages. Nevertheless, nucleic acid bands could get distorted and the gel could be melted down.
Electrophoresis is a very important tool in genetic engineering with several applications, and the technique was learned and applied with running conditions for 120 volts for 40 minutes. The quality of the genomic DNA evaluated was not good when observed with the transilluminator because it was not well stored. It was not a defined band but a degraded DNA. The isoforms of the plasmidic were detected. It was possible to define the size of the PCR product by comparing it with the 1 kilobase DNA ladder. In general, there were also mistakes of protein contamination, loading buffer excess, wrong sample loading. When doing an electrophoresis, it is important to consider appropriate running conditions, percentage and type of matrix, ladder, a color and according to our needs, and the buffer that best suits the electrophoresis objective.